So we're interested in the peripheral nervous system. This is the nervous system that's outside of the brain and the spinal cord and innervates all of the organ systems. And what we're specifically interested in is, after development, you have a peripheral nerve for life. And how does that peripheral nerve stay maintained, and how does it regenerate? And especially we want to understand, as you get older, why does it degenerate? Why do we lose the nervous system, and what are the cellular processes associated with this loss? Why do you lose the nerves? Which means you will get pain. Also motorically you can't move properly, your balance is very bad. And this is associated, what we think, is with a special kind of cell on the peripheral nerve, which I consider to be like the guardian of the nerve itself, which is a glia cell, which is a Schwann cell.
So actually Helen already mentioned some of the most important cells in the nerve, but there are also others, for example the macrophages. And these cells are also very important because all the cells communicate, and they also have to be in a certain interplay in order to fulfill their proper function. And these macrophages sense their microenvironment, and in case of an injury, for example, they become activated and then induce the whole inflammation and regeneration process. They call in more immune cells, and then are important for the whole nerve repair. In order to investigate this, we need proper model systems, and unfortunately we cannot mimic all these processes in a cell culture because, as I mentioned before, we have several cells that are in interplay. Therefore we are using mouse models with certain gene knockouts to mimic disease models, but also just normal mice to investigate aging. So we look at young mice, we look at old mice, we look at their nerves, and we stain for certain markers to see how they look on the structural level, but we also look on gene expression and protein expression.
What I'm mostly working on in the lab is to manage our projects, especially the translational side of it. So we are trying to go a step beyond basic research to get it towards the clinics. One particular cure that we are working on is centered around these Schwann cells. They are really fantastic cells. So they are the guardian of the peripheral nerve. And you see, the peripheral nerve does not have stem cells. Other organs in the body, they can regenerate using stem cells. They make new cells. The peripheral nerve does not have that. They have to get along with the type of cells they have for life. So instead they have the Schwann cells. They are very plastic. They can go from a final stage, which is quite permanent, fulfills the function, to a more immature state, which can promote repair. But this makes it also very dangerous because this repair cell can divide, and if this is uncontrolled, it divides too much, you form tumors. And we're working on a specific molecule that tells them to stop dividing.
Okay, so where do we really want to end up, apart from the details that we work on in the lab and the research that we do? Really where we want to end up is where we actually can prevent degeneration, and we can also prevent tumorigenesis. And why is this important? Obviously, we want to actually promote a long and healthy health span, and we want to preserve the nerves in such that as you get older, you can have a healthy life.